Hello and welcome to another podcast short. My name is Mr. Kelly Carrier and I teach at Okemos High School. My email address is kelly.carrier@okemoschools.net. Today we're going to continue our discussion on cell cycle, and today we're going to discuss sex cells. And in particular, we're going to be talking about spermatogenesis and oogenesis. Now, these two processes are also fall under a broader category called meiosis. Meiosis is much different than mitosis, and one of those differences is that it encompasses two phases, not just one. The ultimate difference is that in mitosis, you will go from a diploid cell, so I will see over here on the far side, a diploid cell, 2N, and then you will get two new cells which are diploid and exactly the same. This is the process of mitosis. Meiosis, however, you will begin with a 2N diploid cell. It will go through the first stage of meiosis, where you will end with a diploid number, and then it will go through its second stage of meiosis, where you will end with a haploid number of cells. And depending on your sex, whether you're a male and perform spermatogenesis or a female and perform oogenesis, this process is slightly different for each of them, and we're going to take a look at it. Again, this is a short, so we're not going to be able to get into all the details, but we'll at least cover some of the basics and some of the things that students often get wrong. First of all, let's begin with discussing spermatogenesis. First and foremost, we're going to begin with a diploid cell. If we were doing our cells, that would mean that we're beginning with a cell that has 46 chromosomes. One of the things that will happen, and often not mentioned, is that these cells will go through the S phase. In other words, the genetic material will duplicate. From this duplication, or because of this duplication, we'll be able to get two new cells with that'll be 2N or have 46 chromosomes. But there's some things that are pretty significant that actually go on here, and I want to talk a little bit about that. First, meiosis occurs in the two stages, meiosis 1 and meiosis 2. Now the nice thing is that meiosis 1 and meiosis 2 also fall into kind of that same pattern that mitosis did in terms of the different phases. It will begin with prophase and end with telophase, and then finally cytokinesis. Let's begin by talking about meiosis 1 right here. The first part of meiosis 1 is going to be prophase 1, and there's some significant things that go on there. They are crossing over and the alignment of homologous chromosomes. Then we'll go on to metaphase 1, where the homologous pairs will align, anaphase 1, where they will separate, telophase 1, where the nucleus reforms for a short period of time, and then cytokinesis occurs and you will get a diploid number of cells. That will then trigger meiosis 2, and we'll talk about that in just a moment. First, prophase 1. There are a couple of characteristics, key characteristic things that go on during prophase 1. They are crossing over and the alignment of the homologous chromosomes. Crossing over. Let's begin there. I'm going to just draw in here a pair of chromosomes, and I'm going to try and talk about both of these things at the same time. Draw in a pair of chromosomes. One will be orange, the other one will be black. Now, one of the things that we're going to try to understand, although it's very difficult because we haven't had a chance to discuss genetics yet, is that each of these chromosomes contains genetic material. Now, for this black one here, this chromosome on the left, chromatid on the left, is exactly the same as the chromatid on the right. And the same thing is true for the orange one. This chromatid here is exactly the same as the one that's over here. Now, what is also true is that these chromosomes, for our purposes, are going to be homologous pairs. So we can indicate that these two are called homologous pairs. Now, the reason that they are homologous pairs is because they contain similar genetic material. Now, for simplicity, and only for simplicity, what we could say is that this chromosome right here came from from the female parent, or your mom, and that chromosome contains lots and lots of genetic information to include hair color, maybe your eyes, and your nose, the shape of your nose. Well, this chromosome over here, which was from the male parent, it will contain similar genetic material. Now, in reality, and for those that are in higher levels of biology, you'll need to know that this is not quite true. That really there's much more complicated, and there's probably more of a blending of different genetic materials that determine certain traits. And so it's not just Mendelian genetics, and it's not this simple. But for simplicity's sake, let's just say that each of these chromosomes has the same kinds of genetic material, so that we have genetic material that's on dad's, which is very similar to genetic material which is on mom's. So by seeing at least that, we can see that these two homologous, meaning that they're the same, they can line up with each other. Now, because of that, there is a significant thing that happens during prophase. It is called crossing over. And crossing over takes a portion of genetic material and will allow it to transfer over to the other chromosome. Now, a very common question is why? Why would that happen? Well, one of the advantages to that is variation, so that now we can have genetic material that it was with, let's say it's a hair color that's with a certain eye color, that now that hair color can be with a different eye color. And again, this is for simplicity's sake, just to try to understand it. Again, the crossing over will take place, and it can take place in either the upper arms or the lower legs. It can take place in a number of different places. And essentially, what it would look like, and how textbooks often portray it, is with the chromosomes actually crossing over, so that a portion crosses over with another one. And then those portions break off, they switch spots, such that you have a portion of the chromosome that is now orange here, and then the other portion of the chromosome which is now black. Something like this. And this is a significant part of prophase, crossing over, and it only occurs in prophase 1. So there's two things that will occur. One is the crossing over, and the other is that the homologous chromosomes will align themselves next to each other. This takes us on to metaphase 1. In metaphase 1, these homologous chromosomes will line up side by side. If we were to look at it a little different like this, they will line up side by side, not in a row, such that we would have, let's say, something like this. Have another set of chromosomes here, slightly different. And this is the homologous pair for this one, and this would be the homologous pair for this one. Now you'll notice that that's vastly different than what it was in mitosis. In mitosis, remember, it was like this, where they lined up like this, so that the chromatids separated into chromosomes. Here, the only thing that happens is that the chromosomes split, where you will finally end up with cells that will have a parent and then the copy of the chromosome. But it will still be considered diploid because it's a double set of chromosomes. So you'll end up with two cells that will have genetic material. If we stay consistent with this one, let's make this orange part up here and make the black part up there so that we can identify. And on this side we will have orange chromosome, and then we'll draw that black in just a moment. We've got the green one that's over here, and we have the teal blue over here, and then I need to put orange in here representing the crossing over, and black over here representing that crossing over, and so that you have two new cells that look like this. Now the next set of steps is in meiosis 2. And meiosis 2 is the most similar to mitosis in that there is no, the homologous pairs don't have to line up because they're in opposite cells now, and they'll go through metaphase, telophase in a line just like they did before when we talked about somatic cells. So these cells will now go on to divide, and where we will end up with something like this, where we'll have a black and we'll have a green like this. On the other side we'll have a black along with a green, and we're also going to put our little orange spot in here. On this portion over here for this cell, it will become haploid, where one of them is orange and the other one is blue. And then on the next haploid cell, we get one that is orange with the black, and we get another one that is blue. I'll make sure I draw the black in here. And one of the things that I want you to be able to see is that not one of these cells are alike. They're all different. So this is haploid, this is haploid, this is haploid, and this is haploid. Now, because we just walked through spermatogenesis, one thing to realize is that these are sperm cells, and they can also be known as gametes. For males, males make sperm cells all the time, while for females, that's a little different. So let's talk about that. Oogenesis is the process for females, and pretty much everything is the same. The crossing over is the same. The crossing over is the same. Homologous chromosomes is the same. Those are the primary things, and the steps are the same. Meiosis 1 and 2. However, there is also one, although there's a lot that's the same, there is one significant difference. Let's talk about that. We will begin with talking about a diploid cell. Again, going through that same process as the other one. S phase will occur, and the first thing that's going to happen is meiosis 1, and it's going to split into two diploid cells. But this is where the similarities end. One of those cells is going to be much bigger than the other. That's not significant enough. Be much bigger than the other, meaning that this cell here has more material, more cytoplasm, organelles, whatever it might be, more material in there, and that is going to help it be more viable, more usable later in its stages. The next set of steps is the same as it would be for meiosis 2. You will still get the chromosomes lining up the same way, like X's, and in a row, and then they will separate. However, again, you will get a separation which will be unequal. You'll get one of them that will be much larger than the other three. Now, what these other three are called are polar bodies. These are called polar bodies, and they are not usable. Often what will happen is that they will get reabsorbed into the body. The genetic material, this could be the absolute best of who you are, but that genetic material will be reabsorbed into the body and used for parts for whatever use that might come by. Well, the only thing that can be used is this cell right here, and usually referred to as an ovum. It is also an egg, and we can also call it a gamete. Now the last step is fertilization. So now we need to take these two gametes and show fertilization. So we're going to take, let's say we take this one right here. So we're going to circle that one. We're going to steal that cell and paste it down here. There it is. That's going to be our sperm cell, and it is going to fertilize this egg. There it is. And once and this is one end. Once the fertilization has taken place, we will end up with a diploid cell. And we call this a zygote. That's the basic process. Now there is one other thing that often throws students off, and that is the idea of the sex chromosomes and how is it that that whole process works. They just get lost, especially when you start talking about the different disorders like non-disjunction, translocation, deletion, inversion, etc. So let me quickly kind of show that idea. Let's say we have a cell that is male, and it's an X and Y chromosome. Realize that what will happen is that this cell will become diploid. All right, so it'll become diploid, and essentially because this genetic material ends up copying, this could be X and then another X, and this could be, let's just for simplicity just put it XY and XY. They will go on and split again, and we will get one cell that has an X in it, one cell that has a Y, one cell that has an X, and the other one that has a Y. The female will be the same idea, but it is XX, and so no matter how it breaks down, it will still only produce a cell that has one X in it. So if we were to take these two and combine them, we would get a boy. If we take, let's say, this one and this one, we would get a girl. And the same thing would be true if we did this one like this. We would again get a girl, and then this one, of course, we would get a boy. And that's kind of the idea and the process that works. All right, and then also notice 50% chance to getting a female, 50% chance of getting a male. There are some other disorders that we also talk about in class, but I will save that for another podcast short. Very quickly, let me kind of review what we just discussed. We discussed the process of meiosis occurring in sex cells. We discussed two processes, spermatogenesis and oogenesis. Both of those will go through the same set of steps with one slight difference. Meiosis 1 with spermatogenesis, prophase 1 crossing over homologous chromosomes, metaphase 1 the homologous pair align, anaphase, telophase, and then cytokinesis, and we end with diploid cells. Meiosis 2 begins, it goes through the process, and then we will end with 4 haploid cells. If it's oogenesis, it's the same stage here, except the one difference is that you'll get one cell that's much larger than the others. That cell will go on to divide just like the other 3, except you will end with one haploid cell that is viable and usable, and you'll get 3 polar bodies. Well, thanks for joining us, and we'll see you next time.